students. How are you, senior two? I hope you are fine. Today we are talking about the photosynthesis process. We discussed before in Zoom session that we have some instructions for the leaves and structure for the chloroplast and what are the components of chloroplast and what are the components of the green leaves. Here today we are talking about the chemical equation for photosynthesis process. Here we need what? We need carbon dioxide, we need water, we need sunlight to get out carbohydrate or sugar and water and oxygen. All of it in presence of chlorophyll. Here guys, oxygen. The one of the scientists which is called Van Nel, an American scientist, he tried to know what is the source of the produced oxygen that produced from photosynthesis process. So here he pointed to the role of the light in photosynthesis process by studying the photosynthesis in green and purple bacteria. So he used to do the experiment. He used green bacteria and purple bacteria. Here he assumed some equations or assumed his experiment in green and purple bacteria that the light decompose the hydrogen sulfide that found in the swamp, which is a purple bacteria leaf, into hydrogen and sulfur in presence of light. And these bacteria reduce hydrogen to reduce carbon dioxide to form carbohydrates in dark reaction while the sulfur is precipitated down. That means that the carbon dioxide reduced from the carbohydrates in the dark reaction and sulfur gets out from this equation. You can indicate that by these equations of the general equations of the photosynthesis process. Here, look, here we have carbon dioxide, we have water, so get out sugar and oxygen and the water. But here in the purple bacteria, we have a carbon dioxide and the hydrogen sulfide, as the swamp filled with hydrogen sulfide, which live in it, the bacteria, so it produce sugar, sulfur instead of oxygen and water. So here the source of oxygen here is from water, not from carbon dioxide. As the carbon dioxide is still here and here, and there is a change here from oxygen to sulfur, as that means that the oxygen source here from water, not carbon dioxide. While in the green plant, the light decomposed into hydrogen and oxygen, as we see here, hydrogen and oxygen, and then the carbon dioxide reduced to form carbohydrates and oxygen release, as you see now, and oxygen release. Okay, guys. So here from Van Nel, we deduce that the oxygen source from water. Green and purple bacteria and also green plant. We can compare between them about the type of chlorophyll here. Here we have a bacteriochlorophyll. Why bacteriochlorophyll? Because it lives in swamp, and this type of chlorophyll is called bacteriochlorophyll. And the green plants, it's called normal chlorophyll, which are A and B. Here the source of hydrogen is H2S, but here the source of hydrogen that the plants need or bacteria need is called H2O. So here the chlorophyll for bacteria is called bacteriochlorophyll, but here for the plant normal A and B. Here source for getting hydrogen is from hydrogen sulfide, then this one that leaves. But here in the green plant we have water. Products for photosynthesis process here, glucose and sulfur, but here is the glucose and oxygen. Here the same is the general equation. Carbon dioxide and hydrogen sulfide get out sulfur that precipitate, and the carbon dioxide reduced by the hydrogen and get out sugar. Here in green plants, no, the water produced oxygen which released, and the carbon dioxide reduced by hydrogen to produce sugar. So we know now from this experiment that the oxygen source is from water, not from hydrogen sulfide, from water, and is assumed by Van Nel on the green algae. Photosynthesis process Here, reactions by Blackman studied that there are limiting factors for photosynthesis, such as light, temperature, carbon dioxide that limited the reaction of photosynthesis process and affected on it. There are two types of photosynthesis reaction, which are light reactions and dark reactions. Light reactions, the site of occurrence for the light reaction, it occurs in grana. You know the grana, remember the grana? Yeah, the grana that found in the thylakoid disc is here. The light reaction occurs inside the during the light. It occurs during the light inside the grana, while during the dark, without any light, it occurs in cytoplasm or stroma. The time of reaction here during the light, but here during the light and they may occur. The limiting factor that affected on the reaction here is the light, but here is the temperature because it doesn't depend on light because of dark. Here in the enzyme not share, but here the dark reaction the enzyme try to share as there is no light that give power or energy for the plant. Here what happened in the process, the kinetic energy, light energy is converted to potential chemical energy and produce carbohydrates inside the chlorophyll, and also the product here is energy ATP, NADH2 and oxygen as second product, and we see the product but after we have finished with this part. After that here we have carbon dioxide which is fixed by the hydrogen that is carried on NADPH2 and help on the energy to be stored in ATP. So here let's start to explain this part again about the light reaction and dark reaction. Light reaction here guys, look, yeah, light reaction, it occurs during the sunlight. The energy used inside the stroma in the light reaction, and the energy is splitting down. Light is split what? The water into hydrogen and oxygen, and the oxygen evolved as a second product, and the oxygen evolved as a second product here. But here look at the hydrogen. What hydrogen of the water? So here the oxygen evolved, yes, oxygen evolved. But here look at the hydrogen, yes, it found NAD. What is the NAD? It's a carrier of the hydrogen that found in the stroma or cytoplasm come to the stroma to grana to get what? To get the hydrogen and the carrier for it to be NADPH2. NADPH2 carrier for the hydrogen to do the dark reaction, as a dark reaction to occur inside the stroma of the green plastid or chloroplast. Here the converting here of ADP, here what happened in the dark, ADP that found also, it found the phosphorus to carry the energy and become ATP that carries the energy to the dark reaction also. So we have energy which change ADP to ATP to do the dark reaction, and also splitting of the light here, also energy carry the hydrogen into NADPH2 to do the dark reaction. So the dark reaction now need what? Need ATP and need NADPH2 to do it inside the stroma. To produce what? To produce something called Bigel. Bigel, yes, Bigel. What is the Bigel? There are 3 carbon atoms that formed due to the reduction of the carbon dioxide and quickly and forming Bigel, and this group of reactions occur in the stroma, and the carbon dioxide gas here is produced after that. So the NADPH2 reduction the carbon dioxide and produce Bigel, which are phosphoglyceraldehydes with 3 carbon atoms. So we need to make a carbohydrate or sugar C6 with 2 Bigel, as the Bigel contains 3 carbon atoms. And the ATP are used in this reaction, yes, are needed in this reaction, and then return back from ATP to ADP to continue again to do light reaction. and so on. They are summary for the light reaction and dark reaction. And the last here experiment that proved that the nature of the dark reaction by Melvin Kelvin at California. He he used the isotope, radioactive isotope, which are carbon 14. He placed the chloroalgae and was supplied with carbon dioxide, but the carbon here is radioactive 14, and the lamp was lighted very briefly to the photosynthesis process, and the chloroalgae here immersed in the beaker with hot alcohol to kill the algae and stop the biochemical reactions that done by it, even if it's photosynthesis process. They separate the product of photosynthesis. He found that what they are bigal. Bigal with 3 carbon atom. Yeah, with carbon 14 radioactive contain 3 carbon atom, which are phosphoglyceraldehyde, which is needed, or it is the second product for the photosynthesis process, and we need to do it by and to make a glucose we need 2 bigal. Okay, guys, so we get the intermediate. Yes, intermediate product is a first stable compound produced from photosynthesis process with 3 carbon atom, and after that we can use it in the cellular respiration. Yeah, look at here is a carbon with the chloroalgae contain carbon 14, and take it. What happened after that? Stop the action quickly to take it and to see what happened. It forms bigal phosphoglyceraldehyde with 3 carbon atoms. I hope you understand this part well. See you soon, inshallah. Goodbye.